expert in the remediation field. We have Dr. Carrie Sublette. Carrie will be giving us an overview of a tool called BioTrap Samplers, and we'll be talking about the diverse application of this tool. Dr. Sublette is a professor of chemical engineering and geosciences and Sarkis Professor of Environmental Engineering at the University of Tulsa. I'm sure many of you know Carrie, and he needs no introduction, but he has over 25 years of experience in a wide variety of bioengineering and soil remediation problems. Carrie also has 13 patents and over 117 publications ranging from corrosion to brine spill remediation to ecosystem restoration. He chairs the annual International Petroleum Environmental Conference, and he also serves on the Oklahoma Board of Environmental Quality. As many of you who have attended before know, this webinar today will be recorded and available on the resource section of the Microbial Insights website within the next week. We'll also email a link to all registrants as soon as it's available. At the end of this morning's webinar, there's going to be a question and answer period, so please go ahead and type your questions into the GoToWebinar software, and Carrie will answer them at the end of the presentation. If you have additional questions after the webinar, you can also email them to info@microbe.com, and we'll get with Carrie and get those answers for you and get back to you. So without any further delay, I'll go ahead and turn it over to you, Carrie.
Thank you, Dora. As Dora said, what we'll be talking about today are applications of biotraps, particularly for environmental site diagnostics. And for those of you that aren't familiar, I'm sure the first question you have is, what is a biotrap? Basically, a biotrap is a passive sampling tool for microorganisms. Passive in that primarily we might deploy a biotrap in the groundwater monitoring well, but when we deploy that biotrap in the groundwater monitoring well, we simply leave it there typically for about 30 days or so, and then retrieve it for analysis. There's no care and feeding that goes on during the incubation period. It's simply there, and that's why we refer to it as being passive. The most important thing to remember about a biotrap sampler, and I'm going to illustrate this as we go along, is what it collects. Because it collects only active microorganisms in the sampled environment. And by that I mean this: for a microorganism to be detected in the medium that makes up the biotrap, called the bioset bead, which I'll be describing momentarily, microorganisms have to get on the inside of that bead, and once inside they need to grow and reproduce in sufficient numbers to be detected. So if an organism is not actively growing and reproducing in the sampled environment, you will not detect it in the beads. So in that way, the biotrap is helping you eliminate all this microbial noise that exists in any natural environment and allowing you to focus strictly on those organisms that are actually actively involved in the microbial ecology of the site. Another thing to consider with a biotrap, or to know about a biotrap, is that it's an integrated sample that we take, not a snapshot sample. When we deploy a biotrap in a groundwater monitoring well, for example, and it's incubated there 30 days or so, the biofilms that are formed on the inside of these beads then are coming into equilibrium with the sampled environment. So we're basically providing an integrated sample versus a snapshot, where a snapshot might be going out and simply taking a groundwater sample and filtering it and sending that off to the laboratory for analysis. As we all know, the ecology and the chemistry of a groundwater monitoring well can change over time. So a sample that we take today may not be the same as a sample we take 2 days from now, or 3 days from now, or 10 days from now. So by taking an integrated sample, we're dampening out those fluctuations that naturally occur in both the chemistry and the microbiology of those groundwater monitoring wells over time. Now, once we've collected the microorganisms, as I'll be illustrating for you, we can analyze the beads in the biotrap and the microbial cultures we've collected using a variety of different kinds of molecular biological tools. We could also do analysis of just chemical methods as well as stable isotope analysis. But I'll be illustrating that as we go along. So how does a biotrap work? Well, the secret of the biotrap is a particular type of collection medium called a bioset bead. And you can see a low magnification cross-section of a bioset bead in the upper right-hand corner of this slide. These beads are about 3 to 4 millimeters in diameter, about the size of a BB, and they're composed of only two materials, Nomex and powdered activated carbon. Nomex, you might recognize, is the polymer that's used to make fire retardant suits and first responder suits. So it's a very temperature tolerant organic polymer. And as I said, the rest of the composition of the bioset bead is powdered activated carbon. So you can kind of think of the Nomex as simply the matrix that's holding the powdered activated carbon particles in place. Now, the nature of this bead is that of one of very high porosity. About 74% of the inside of the bioset bead is empty space, but there's a lot of surface area, on the order of 600 square meters of surface area per gram of bead. The other two photos you see on the right-hand side, the middle one is a high magnification SEM picture of a microbial biofilm on the inside of a bioset bead after incubation, and then the photograph in the lower right-hand corner is a live dead stain, again, of a biofilm on the inside of a bioset bead. Now, one of the properties of these beads, since the Nomex is so heat tolerant, is that these beads can be heat sterilized up to 270 degrees C. So the big advantage here is that not only can we sterilize these beads, but since we can preheat them to such high temperatures, we can basically drive off any organics that are adsorbed to the activated carbon and basically burn off or destroy any biomarkers that may be present in the bead. So that when you get a biotrap, the bioset beads in that biotrap have been preheated to 270 degrees C, and so you can be sure that those beads are clean and sterile before they go down into your sampled environment. So anything that you find on the inside of the beads after incubation, you can be sure comes from your environment. And as we've already said, the bioset beads are colonized by active microorganisms. So what kinds of forms do the bioset samplers come in? And the short answer to that is any way you like it. But there are some standard biotrap designs. This is one of those standard biotrap designs. That biotrap that you're looking at there is about 4 inches long and about a ½ inch in diameter, and it's made out of PVC. And is packed with about 250 bioset beads. This is kind of the standard design that's used in groundwater sampling, but we've also buried them in soil, buried them in sediments, any kind of environment you can imagine. We can place that particular trap. Now, the second type of common biotrap sampler is the one you see here. This is an in situ microcosm. This is a device marketed by Microbial Insights, and I'll be giving you some examples of the use of this sampler. But this sampler, within that sampler, as you can tell on the right-hand side, we have several things, one of which is a biotrap. But we can also place inside that in situ microcosm other types of samplers too, with passive diffusive membranes to monitor geochemistry or dissolved gases, etc., and other types of sampling devices also with different passive membranes to sample organics like petroleum hydrocarbons or chlorinated hydrocarbons, etc. Typically, these are going to be deployed in groups like the one that you see here. One is marked M&A for monitor natural attenuation, and then we have two that are marked treatment option 1 and treatment option 2. And as you'll see in some of the examples that I'm going to give, basically what we do in the treatment options is we alter the environment on the inside of these in situ microcosms by adding electron acceptors, electron donors, doing bioaugmentation, various kinds of things, as I'll illustrate. Here is just a little dye test just to illustrate the way these in situ microcosms are isolated from each other. If you look at that previous slide, you can see the baffles on the end of each one of these units to make sure that they are hydraulically isolated from each other in the groundwater monitoring well. The various types of electron donors, electron acceptors, this is just a partial list for illustrative purposes. And a little bit later I'm going to tell you about a technique called stable isotope probing. And we can also put various C13 labeled compounds in these in situ microcosms as well. So the biotraps in the in situ microcosms and the standard biotraps are the two most common. How are these biotraps going to be deployed? Well, typically, as I said, the most common environment these are deployed in is a groundwater monitoring well. And typical deployment would start with purging the monitoring well, and then you would suspend the biotrap or the in situ microcosm from the top of the casing, and typically you're going to deploy that biotrap in the center of the screened interval. Okay? Or if there's a particular vertical interval that you're interested in, of course you can position the biotrap in that specific position as well. In certain cases, if there are large fluctuations in the water level that are anticipated, you can even suspend the biotrap from a float, so it will rise and fall with the water table and maintain its position with respect to the water table. Where do we deploy biotraps? Well, in a typical contaminated aquifer, we might consider a deployment scheme something like this, deploying a background trap and then perhaps through the axis of the spill, and depending upon resources, we might also be interested not only in the fringes of the spill and what's going on there, but also looking down gradient as well. If you're interested in sampling over time, there are a couple of different strategies for doing that. You could deploy a biotrap today, for example, then retrieve it 30 days later and replace it with a fresh biotrap and continue that process during the entire time that you want to sample. Another option is to deploy several biotraps at once and then simply retrieve them over time. And illustrated here is a 30, 60, and 90-day retrieval. Now, those are the most common biotrap designs, but I don't want to leave you with the impression that that's the only type of biotrap that has been used. There are many, many different types of specialized designs. For example, here is a flow-through biotrap, this particular one being used on a slipstream and a drinking water distribution system. Here is a biotrap assembly that was used to sample below a hydrocarbon layer in the aqueous phase in a solvent extraction system. Here are some very small biotraps that you might recognize that as a coupon holder. This was deployed in a pipeline in Alaska monitoring microorganisms. Here's another type of biotrap design that in this particular case the client wanted to directly sparge the biotraps and to change the equilibrium dissolved oxygen concentration in the biotraps by sparging each one of the biotraps with a different partial pressure of oxygen in the sparging gas. And then here is another design. This one was used actually offshore in China, using a piezometer as a housing. You can see the biotrap there. It's a bit longer in this case than the standard biotrap, and it was deployed adjacent to the slots that you see in the piezometer. And then the entire assembly was basically hammered into the sediments on the ocean floor at the desired depth. And then lastly, here are some biotraps. Some are standard biotraps. Some were actually amended with hydrocarbon. These particular biotraps are deployed at 5,000 feet in the Gulf of Mexico. This was during the BP Gulf oil spill. So there are lots of different types of biotraps that can be deployed for lots of different reasons. Once the biotraps are retrieved, as I said earlier, we can do chemical analysis. We can use molecular biological analysis. Here's just an example of some of the types of things that can be done. The beads, for example, can be extracted for lipids. They can be extracted for DNA. They can be extracted for stable isotope compounds, and then use PLFA analysis, qPCR, quant arrays, DGGE, any of those types of molecular biological tools. We can even extract adsorbed organics out of the beads and do compound-specific isotope analysis. We can also analyze dissolved inorganic carbon, and then, of course, any contaminant concentrations that are going to show up either in the aqueous phase or the adsorbed phase on the inside of the beads. So what can I do with a biotrap sampler? Well, one thing I can do is simply determine if known degraders of a contaminant of concern are present in a sampled environment. Now, one of the organisms that people are always analyzing for when it comes to chlorinated hydrocarbon sites, for example, are Dihalococoides. But Dihalococoides has no known mode of motility. So you might wonder how an organism like this might be collected with the Bioset beads. But as you can see, they certainly are. This is from Microbial Insights database, looking at thousands of different samples and the distributions of, in those samples, the concentrations of Dihalocoides that were detected. And so obviously, simple Brownian motion is sufficient to get these organisms on the inside of the bead. And then once they get on the inside of the bead, then they will grow and reproduce there if the conditions in the aquifer are favorable to the growth of the organisms. Just another sample here. These are hydrocarbon degraders. These are particularly looking at catabolic genes related to hydrocarbon degradation under aerobic conditions, and you can see the distribution here. And one of the things that might be of interest to you is to look at zero. How often the answer is the organisms aren't there, which might be curious to you because we all know that hydrocarbon degraders are ubiquitous in the environment. We're always going to be finding them. So why do we not always find them in the beads? And the answer lies in what I was referring to earlier about how or what Bioset beads collect. They only collect the organisms that are actively growing and reproducing in the environment. So in those samples, we know the hydrocarbon degraders were there, but for one reason or another, maybe there was a missing nutrient, maybe the environmental conditions were not right, they were not growing and reproducing, and if they're not growing and reproducing, they will not show up in the biotrap. So again, in that way, we're filtering out all that microbial noise and focusing only on organisms that are actively involved in the microbial ecology. And to just show you a little bit more field data now, here we're looking at a particular site where we have biotraps in 2 different monitoring wells in an aquifer impacted by chlorinated hydrocarbons. And as you can see in this example, Dihalococoides was detected, as well as vinyl chloride reductase also detected. So going on down our list then of what can we do with a biotrap sampler, we have 2 bullet points that are very much related to each other. One, we can evaluate monitored natural attenuation versus enhanced bioremediation. And going along with that enhanced bioremediation, we can compare the effectiveness of amendments that are designed to stimulate bioremediation. Now, very frequently, when we employ a biotrap to demonstrate either or both of those points, we're going to use an in situ microcosm, as I described it a few minutes ago. But just to give you a little bit more information now, there again is the biotrap in the center. Above that, we have a sampler marked COC. Below that, a sampler marked GEO. And below that, basically a sponge device called supplier. Again, with the biotrap, we can extract DNA, lipids, whatever we want out of that biotrap. If it's DNA, we can do qPCR, quant arrays, etc. And We can also do stable isotope probing, which I'll be describing in more detail shortly. In the top sampler, we can sample for VOCs, chlorinated hydrocarbons, any sort of organic that's present, any contaminant of concern that's present in the aquifer can be sampled. The sampler marked GEO, any cations, anions help us understand redox conditions. We can look for dissolved gases like ethene, ethane, etc. So if we're looking at a hydrocarbon impacted site, how might we deploy these in situ microcosms? Well, a common type of deployment might look like this, where we have a control and two that were labeled here biostem. The M&A is always going to be the unamended microcosm. So basically when we look at the conditions that we find there, both microbially and chemically, what you're looking at basically is the baseline condition. This is what the chemistry looks like. This is what the microbiology looks like in the absence of a remediation amendment. However, in the two treatment options, in this case one labeled oxygen, the other labeled sulfate, either oxygen coming from an oxygen release compound, or in this case, sulfate as a terminal electron acceptor. And what the analysis of those two in situ microcosms will then tell us what will happen to the chemistry, what will happen to the microbiology if oxygen is supplied, or if sulfate is supplied. If we're talking about a chlorinated hydrocarbon site, this might be a possible scenario for the in situ microcosms. Again, we have an M&A microcosm. Again, that's given us the baseline condition, the chemistry, the microbiology, in the absence of any remediation options. In this particular case, we've got one that's labeled biostem, and that might be, for example, an in situ microcosm in which we amend it with some sort of electron donor, lactate, HRC, EOS. There's numerous examples of that, as you know. The third one here, we're labeling bio-aug, and in the bio-aug, this might be a unit, and I see there's a typo here. That should not be sulfate. But in a bio-aug unit, basically we're going to supply an electron donor, and we're going to bio-augment the in situ microcosm with a commercial culture. There are several commercial cultures, as you know, that are marketed for chlorinated hydrocarbons. It is possible for us to take the biotrap in one of these in situ microcosms and preload it with that bio-augmentation culture, in addition to an electron donor. So with this, we can determine that will that bio-augmentation culture, number one, survive in that environment, and number two, will we see an effect on both the microbiology and the chemistry when these units are supplied. And one thing that I should point out about these in situ microcosms and these electron donors that are present, these are very small amounts of electron donors. They're meant to change the environment on the inside of these in situ microcosms. After incubation, there's very little or none of this electron donor that's actually left. There is no lasting effect of that electron donor, especially after any subsequent purging when the well is being resampled. So I'd like to show you a case study using in situ microcosms and biotraps at a chlorinated solvent site. This particular site, the primary contaminant was TCE. Daughter products in the form of cis-DCE were detected, but no vinyl chloride, no ethene, so it seemed to be, the standard DCE stall. So the client in this case wanted to look at biostimulation, electron donor addition, as well as possibly bioaugmentation, so using electron donor and a commercial culture. So the questions to be answered here in terms of the microbiology are, number one, we'd like to know, are the organisms capable of complete reductive dechlorination of TCE to ethene, in other words, dihelicocoides? Are they present under M&A conditions? Further questions, will the addition of electron donors stimulate the growth of these organisms? Of course, if they're not there, electron donors has nothing to stimulate. Will a bioaugmentation culture survive? And the big question, of course, is then if it survives, is bioaugmentation necessary at this particular site? With respect to the chemistry, we want to know, will this donor addition promote daughter product formation? In other words, can we go past cis-DCE and stimulate further reductive dechlorination? We also want to know then, of course, if bioaugmentation is going to be more effective in enhancing reductive dechlorination than the electron donor addition alone. So the study design looked like this. It's basically the standard type of train of in situ microcosms that we've gone through. And again, we'll use the COC samplers to look at the TCE and its daughter products. We'll use the biotrap to look for dihelicocoides as well as genes involved in reductive dechlorination like vinyl chloride reductase. And we'll use the geosampler to look for dissolved gases, in particular ethene production. In the biostem, the suppliers will be used to supply the electron donor. In the bioaugmented, again, the biotrap will be pre-incubated with the commercial culture to load that commercial culture into the beads before incubation in the monitoring well. So let's look at some results. First of all, in the M&A in situ microcosm, as you can see here, the dihelicocoides was detected, low concentrations, but it's there, and vinyl chloride reductase was detected. So those questions we were asking, is dihelicocoides present? Yes, it's there, and we're also detecting the presence of vinyl chloride reductase genes. Next, we look at the biostimulation in situ microcosm, and we see that compared to the M&A, we see an order of magnitude stimulation and the concentrations of dihelicocoides. We also see a stimulation and the concentration of vinyl chloride reductase genes. And now we're also being able to detect TCE reductase genes as well. So remember that original question, will the additional electron donor stimulate growth of these organisms? The answer is definitely yes. Now if we go on and look at the bioaugmentation unit, as you might expect, you see a much higher concentration of dihelicocoides in these bioaugmented in situ microcosms. And that's exactly what we'd expect if the bioaugmentation culture that we previously loaded into the beads is going to survive in that environment, and certainly it did. And as well, we're seeing high concentrations of TCE reductase as well as vinyl chloride reductase. So again, the question, will the culture survive? Yes, it certainly did survive. Is bioaugmentation necessary? Well, let's revisit that question when we see the COC results. So here we're comparing the COC results from the COC sampler in the M&A in situ microcosm and the biostimulation in situ microcosm. As you can see, in the absence of an electron donor, we're detecting both TCE and 1,2-DCE as expected. Now, in the biostimulation unit, one of the things that we see is that there has been a very significant decrease in the concentration of TCE, and a lot more of the TCE has been converted to DCE. But we're also picking up traces now of vinyl chloride and ethene. So remember, one of those questions was, will the donor addition promote a daughter product formation and stimulate complete reductive dechlorination? The answer seems to be yes. Now, if we compare that to the COC results from the bioaugmentation culture, now we see the TCE is totally gone. We are seeing slightly higher concentrations of vinyl chloride and higher concentrations of ethene. But the results are not staggeringly different. So the question was, will bioaugmentation more effectively enhance reductive dechlorination than the electron donor addition alone? Well, the answer seems to be yes, but will bioaugmentation be necessary? The answer is probably not. We can probably get higher rates with bioaugmentation, but the question remains as to whether that would be cost effective, because with electron donor alone, we can get complete reductive dechlorination. Here is another beaded biotrap type of study. This is a gasoline-impacted aquifer. Now switching to hydrocarbons, a shallow aquifer impacted by gasoline, of course, BTEX components were the contaminants of concern. Dissolved oxygen concentrations were typically quite low. Concentrations of other electron acceptors also fairly low. The client in this case was considering oxygen addition to stimulate BTEX biodegradation. So, of course, our site-specific question here: is oxygen addition going to promote the growth of the organisms that are capable of aerobic degradation of BTEX? So once again, we use an in situ microcosm. It looks very similar to the one you've seen before. Again, there's a sampler for COCs, for geochemistry, there's a biotrap, and then there's a sampler for oxygen, a supplier for oxygen, in this case, an oxygen release compound to supply oxygen in the in situ microcosm. So just to show you quickly some of these results, looking at 3 different genes involved in aerobic hydrocarbon degradation: phenol hydroxylase, toluene dioxygenase, xylene monooxygenase. Here in each case, we're comparing the control, or the MNA, compared to the oxygen amended. And what we see, first of all, looking at phenol hydroxylase, is a 2 order of magnitude increase in the concentration of those genes, well over a 2 order of magnitude increase in the concentration of toluene dioxygenase genes. And for xylene monooxygenase, that was non-detect in the MNA in situ microcosm, and was detected at moderate concentrations in the oxygen amended in situ microcosm. So obviously, the addition of oxygen is going to stimulate the production of those genes, or the expression of those genes that are involved in aerobic hydrocarbon degradation. So moving on now to the last bullet point, we want to prove that bioremediation of a specific compound is occurring under in situ conditions. To do this, we're going to use that technique I referred to earlier, stable isotope probing, or SIP. So what is stable isotope probing? Stable isotope probing is a method by which we use stable isotopes to coupled with molecular biological tools that, as you will see here shortly, can prove the biodegradation potential of a particular target compound under actual in situ conditions. And further, we can actually link the biodegradation using stable isotopes to the organisms that are actually responsible for the degradation process. So if you're unfamiliar with stable isotopes, the particular one we're interested in is carbon-13. Naturally, carbon-13 makes up about 1% of all the carbon in the natural world. But what we're going to do is we're going to use contaminants of concern. The one shown here is benzene, that have been synthesized out completely with carbon-13. The compound, the only difference between, say, benzene that is fabricated totally from carbon-13 and natural benzene is just going to be in mass. They're going to have the same chemical and physical properties, except for those that are going to be affected by mass. What we're going to do is to use that carbon-13 as a tracer to help increase our understanding of what the fate of a particular contaminant is going to be in a particular environment. Now, I want to make a distinction between a technique that you might already be familiar with, compound-specific isotope analysis, and stable isotope probing. Both focus on carbon-13, but they're very different. In compound-specific isotope analysis, we're looking at the natural isotopic fractionation that results when a carbon compound undergoes biodegradation and carbon-carbon bonds are cleaved. Bonds involving carbon-12 and carbon-13 basically cleave at slightly different rates. The result is that as biodegradation proceeds, then we see a change in the carbon-13, carbon-12 ratio as that biodegradation proceeds. But these changes are quite small, and it takes very sensitive instruments to detect them. In stable isotope probing, as you're going to see, we're basically talking about stable isotopes with a hammer. We're going to use carbon-13 simply as a tracer. As shown in the diagram on the left-hand side, and I'll illustrate this more, we're looking at the movement of carbon-13 into the products of mineralization, like carbon dioxide and methane, and we're looking at the incorporation of carbon-13 into biomass following biodegradation, into phospholipids, into DNA, in proteins, or whatever you would like to analyze. So basically, this is the way it's going to work. Here's a biosep bead. Remember, the biosep bead is made up of 75% powdered activated carbon. We're going to load a C13 labeled contaminant, here we're showing benzene again, into the bead. This is done typically in the vapor phase and results in adsorption of the compound on the activated carbon component. But I'd like to stress that there's more than activated carbon adsorption going on here. Because of the pore size distribution, there's two forces that are basically holding that compound inside the bead. One is the adsorptive properties of the powdered activated carbon, but the other is simply the mass transfer resistances now that are going to exist when this compound gets adsorbed deep into those pores. So once we have loaded the compound on the activated carbon, we can then deploy that bead in an environment where there might be microorganisms that are capable of utilizing that carbon compound as a carbon and energy source. So those organisms and any organisms that can grow in that environment are going to colonize that bead. And those organisms that are capable of degrading that compound will do so. And as they do so, if the carbon-13 is indicated as it is here in red, you'll see that a couple of things are going to happen. One is some of these organisms are turning red, as you can see, to illustrate that C13 from that contaminant has been incorporated into the biomass of these microorganisms as they grow and reproduce. You also see some of the symbols here for carbon dioxide are red to indicate that some of this compound is being totally mineralized, and when it is, part of that is going to go into CO2. All of it's going to go to CO2 if it's aerobic. If it's anaerobic, you'll actually see both CO2 being labeled as well as methane being labeled as well. So just to illustrate the process again in one slide, we take a C13 labeled compound here, benzene, is loaded onto the beads. The beads are then placed into a biotrap. The biotrap is placed into the environment that we wish to sample, typically a groundwater monitoring well. After incubation, the biotrap is retrieved, and the beads are retrieved, and the beads are then extracted for analysis. Now, a question that may be coming to your mind is what happens to that adsorbed compound during incubation? Does it stay in the bead, or does it leach off the beads? Well, when it comes to hydrocarbons and similar types of compounds, The absorptive properties are such that those hydrocarbons do not leach off the beads hardly at all. Anything that we do actually lose from the bead is very minuscule. As you can see here, this is a leaching study that was done with benzene dissolved biosat beads, and you can see 95% of the benzene is still there after 30 days of incubation. And similar results for compounds like xylene, toluene, naphthalene, etc. In fact, naphthalene sticks like glue. Phenol sticks extremely well. Other compounds that we might be interested that may contain oxygen do not adhere quite as well. But what things like MTBE, T-butyl alcohol, for example, are the ones that come to mind. They will be lost from the bead to some extent, but our analysis has shown that basically when we load those types of compounds onto biosat beads, that the contaminant is there basically in two types of pools, a kind of a labile pool and a pool that is held much more tightly. So to prevent significant leaching during the incubation of these types of amended beads, what we will do is to basically pre-leach the bead for 5 days and in sterile water, and after 5 days that labile pool that was there is now gone, and what you're left with is the pool of compound that is absorbed more tightly. So that if you use any one of these types of compounds in a SIP process, you can be sure that Microbial Insights will be doing this type of pre-leaching before they ever come to you. Okay, well, once we retrieve the compounds, then what kind of analysis do we do? Well, there are basically three types of analyses. One, we want to look at the amount of the residual compound that's still left in the beads. When they come to you, Microbial Insights knows exactly what the concentration of that compound was in the bead when it went to you. One of the first things they will do is they'll look to see what the concentration is when it comes back. You know, is there 10% loss, 20% loss, 50% loss, etc. This is a potentially useful piece of information for you, in that if you are deploying biotraps in multiple wells at a single site, the amount of loss of that compound can give you an idea of the relative rates of degradation of that compound at your site. Now, don't get excited. You cannot get a rate constant from this. And I want to emphasize that all we're doing here is comparing this part of this aquifer to that part of this aquifer. If we're getting 90% loss over here, but we're only getting 40% loss over there, then you can be pretty confident that you're getting greater rates of that target compound where you saw the 90% loss compared to where you saw the 50% loss. All right? But that's why I've underlined here, this is a local comparison, and it's simply a relative comparison, comparing one well to another at a single site. The other types of analysis we do, we can extract for dissolved inorganic carbon, and if it's anaerobic conditions, we can also extract methane as well. But we'll always extract dissolved inorganic carbon, and we'll look at the C13-C12 ratio in that dissolved inorganic carbon. But we can also look for C13 incorporation into biomarkers. Now, the most common biomarker to be analyzed will be phospholipid fatty acids. And the only reason for that is because there's such a high concentration of phospholipid fatty acids in microorganisms. So if we're simply wanting to detect the incorporation of C13 into biomarkers, that's the go-to biomarker. But if you are interested, we can also look for C13 incorporation into DNA, and we look for C13 incorporation into RNA as well. As I'll show you an example, looking at how the C13 is distributed in phospholipids can give us an idea of what kinds of organisms are involved in that cycling process for that compound. If you look at C13 incorporation into DNA, we can potentially identify the actual organisms that are involved in the degradation process. If you're not familiar with phospholipid fatty acid analysis, phospholipids make up the majority of the cytoplasmic membrane. There are about 14 to 16 of these common phospholipids, and the C13 will then get incorporated into these phospholipids. If you can look in the upper right-hand corner there, you see kind of a cartoon drawing of a phospholipid with a polar head and two nonpolar tails. As I said, those nonpolar tails, they're about 14 to 16 common structural varieties of those, and as I'll illustrate for you a bit later, that can give you some idea of what types of organisms that you're looking at here. If you determine that C13 has been incorporated into certain kinds of lipids or incorporated at greater amounts in certain kinds of lipids, it then gives you information about which organisms are more involved in the cycling of that C13. But I'll illustrate that later. I just wanted to illustrate here on Microbial Insight's website, which is given here. You can find a lot of information about the different structural types of phospholipids and what kinds of organisms typically contain high concentrations of those types of phospholipids. But I'll illustrate that more a bit later. Now I want to show you some results, but before we do, I want to make sure that we all understand how we talk about the C13-C12 ratio in an organic compound. We do that in terms of the del 13C notation. Basically, it's a calculation based on the C13-C12 ratio in a sample compared to that of a standard, the standard being PD belemnite, and it's calculated by the equation that you see here. If you perform that calculation, it results in a scale like the one that I've shown you here, where the standard then is going to have a del 13C value of 0. So anything that's any compound that's going to have a del 13C value that is less than 0 contains less carbon 13 than the standard, and if it has a value higher than that, that's more carbon 13 than the standard. What you're looking at here are some common contaminants, things like benzene, some chlorinateds, MTBE, and what the del 13C values of those compounds typically are, as either they're found in nature or produced industrially. Now when we do stable isotope analysis, we're going to be talking about del 13C values that are basically way off this chart, highly positive values, indicating very clear results that the C13 compound of C13 content rather of the biomass, the molecular biomarkers or products of mineralization are much, much higher than would be normally found in nature and will be proof positive of the incorporation of carbon 13. So let's look at a couple of examples. The first example, this is looking at the feasibility of M&A at a former manufactured gas plant. This is a site map. It's not very meaningful for our discussion here, but the common contaminants here, or the most common contaminants, are benzene, ethylbenzene, and some polyaromatics, like some naphthalene. Just to show you some concentrations here, I'm going to focus on the naphthalene and 2-methyl-naphthalene at different wells on the site, as well as we've got some background wells here too. Looking at the trends over time in the contaminant concentrations, again, I'm going to focus on naphthalene. You're seeing, in some cases, you're seeing decreasing naphthalene concentrations. In some cases, you're not seeing any trend. And in one case there, we're near the detection limit. So there is some evidence from the contaminant concentration that perhaps naphthalene is being biodegraded. But we would like to add to that by doing stable isotope probing and also doing some qPCR using a quantarray to look at multiple catabolic genes to add some more evidence to the evidence that we have here. If you're not familiar with the quantarray, it's simply a method, and you can get more information about this from MI's website. um, of doing multiple qPCR analyses simultaneously. In this particular case, looking at many different catabolic genes for aerobic and anaerobic degradation of hydrocarbons, including naphthalenes. So just to show you some results, what was done at this site was biotraps were deployed that had been loaded with C13 labeled naphthalene. And here you're looking at one of these wells, and as you can see the del13C value, this is for the DIC, the CO2. Look at the values of these DIC values. They're quite high. Remember the normal scale, the normal concentration, background concentration for carbon dioxide in these environments is going to be somewhere around about minus 25. And here we're dealing with, in one case here, about plus 200. So there is no doubt that naphthalene has been biodegraded, and some of it has been mineralized all the way to CO2. Looking at results from the same well, now looking at C13 incorporation into biomass, in this particular case into lipids. And again, background would be very, very low, but again about minus 25, minus 30 or so. Here we're seeing values above 750, plus 750. So again, there is no misinterpreting what you're looking at there. You're looking at C13 incorporation into biomass. So naphthalene, originally loaded with C13, supplied to the culture, so to the organisms in this environment. You see incorporation of C13 into biomass. You see incorporation of C13 into carbon dioxide. That is proof positive that organisms capable of naphthalene biodegradation exist in this environment and did degrade that compound under the actual conditions that exist in that environment. Here's some quantarray results showing the detection of both aerobic genes involved in naphthalene degradation as well as anaerobic genes involved in naphthalene biodegradation. So in assessing the degradation of naphthalene at the site, particularly M&A, the chemical analysis does show some decreasing contaminant concentrations. That's a green light. The stable isotope probing definitely shows that naphthalene biodegradation is going on. That's a green light. And then there are multiple genes from both on the anaerobic side and the aerobic side known to be involved in the degradation of polyaromatic hydrocarbons also detected. So we've got green lights all the way across the board indicating that naphthalene biodegradation is definitely going on. And I'm going to quickly end with another SIP study. This one, and I'll just go to the final slide here on the site. This is a study of a paint manufacturing plant looking at xylene. And the client here wanted to know, can we stimulate xylene biodegradation, this paraxylene, using aeration. So we got 3 wells here where aeration took place and 1 well where it did not. And again, C13 labeled xylene was loaded into these biotraps, incubated for 30 days, and then analyzed for C13 incorporation into dissolved inorganic carbon as well as biomass. So here is some results for dissolved inorganic carbon. Extremely positive values. There is some degradation going on without the injection of oxygen, greatly stimulated with oxygen, and it's the same story looking at C13 incorporation into biomass. Very, very high positive values for lipids in the areas that where oxygen was being injected. But the primary thing I wanted to show you was this. In this particular case, here you're looking at a number of common fatty acids that are found in those phospholipid fatty acids. This is basically the control where the concentration of xylene was about 10 parts per million, and it was not aerated. And what that line shows right there is the background C13 concentration. This is an atomic percent of C13. So if in looking at these individual fatty acids, if that bar extends above that line, then there was C13 incorporation. And as you can see, there was a small amount of C13 incorporation into several of those fatty acids where in the outside of the aeration zone. Now within the aeration zone, as you can see as I go to several of these samples here, quite a bit more C13 being incorporated into these various fatty acids. Now what I wanted to show you though was this. Remember I said you can find information on the MI website relating some of these different structural classes of fatty acids to different kinds of microorganisms. For example, the first group that you see here are very typical of anaerobes. The second group here you see are very typical of aerobes. And you can see in this particular case there was C13 incorporation into a particular fatty acid that is characteristic of certain sulfate-reducing bacteria. And in the right-hand side there, you see a fatty acid that is typical of fungi. So you can get a picture then of what types of organisms are incorporating most C13. And in this particular case, what you can see here, and this is pretty typical with introduction of oxygen into a hydrocarbon environment, that you're going to stimulate, excuse me, stimulate not only the aerobes, but you're also going to stimulate the growth of many, many different types of microorganisms, including anaerobes. And as you can see, that C13 that comes from that xylene is being cycled and incorporated into the biomass of a variety of different organisms. I want to also refer you to a resource on the Microbial Insights website. This is a SIP database where they keep track for different contaminants of concern of what kinds of incorporation into CO2, what kinds of incorporation into biomass are typically seen for these various COCs. So with that, I will stop and answer questions, but you can get a copy of this presentation if you'd like, either from Dora or myself. Just email either one of us, and we will send you a copy.
Well, thank you very much, Kerry, for the presentation. It's an excellent overview of all the tools and the way we can use the BioTrap samplers. So the first question we have here is, has anyone done a side-by-side in situ test of a BioTrap versus plain water samples to be able to compare as far as detecting and quantifying a particular organism or genes and how that looks different in a soil or a groundwater and a BioTrap sampler?
Well, excuse me. If I understand the question, I did a study once at a site impacted by basically gas condensate liquids, in which I looked at analysis of water samples, and I looked at samples with BioTrap, taken with BioTrap. So we're talking about integrated BioTrap samples versus just snapshot type samples. And I had two zones, multiple wells in two zones, a control zone and the impacted zone. And what I was able to show was that looking at the microbiology in the biotraps, I could distinctly tell the difference between these two zones. However, it was statistically difficult to distinguish the two zones by taking just the snapshot samples. Now, I'm not going to say that's going to be that way all the time, but what it really shows in my mind is the integrated approach that smooths out the variation that occurs on a daily basis in a lot of these sites. I think the integrated approach gives you a much better picture of actually what's going on at the site.
Okay. The next question, Kerry, is how do cell concentrations per bead compare to cell concentrations per liter?
Well, I don't necessarily know exactly what that relationship is. However, there will be a relationship on a site-by-site basis, because either the microorganisms are getting into the beads by some means of motility, or they're getting in by Brownian motion, But in both cases, the getting into the bead, the rate at which they get, or frequency, I guess we should say, at which they're getting into the bead is going to be directly dependent upon the concentration of those organisms in the aqueous phase. But I can't tell you, for example, that a concentration in cells per bead in this aquifer means this concentration in cells per milliliter. We'd have to do some comparison on a site-by-site basis to determine that. What I can be sure of is that there is a relationship at every site.
Okay. You also mentioned that biotraps are good for Dihalococoides because of the Brownian motion. Are there other types of bacteria or archaea that you're worried about that may not be suitable in the biotrap, and perhaps something that's a more filamentous organism?
Filamentous organism, that's a good question. I have definitely, when I've done SEMs of bioset beads in the past, I have definitely seen filamentous organisms on the inside of the bead. But whether they got there by growing into the bead as a filament or whether they got there as a spore, I don't know. But my guess is that it was one of those two mechanisms. An organism as a long filament probably is not going to get into the bead by Brownian motion unless it simply grows into it. We have definitely seen that happen, though.
Okay. The next question is, 1,4-dioxane is a compound of concern right now that people are evaluating. What's your suggestion for applying a biotrap to 1,4-dioxane evaluation?
Well, it's definitely been done and definitely been done successfully. Because 1,4-dioxane does not adhere as well to activated carbon as does a pure hydrocarbon, it is best to go through that pre-leaching phase that I referred to earlier. But having done so then and deployed into a groundwater environment, we have several times seen C13 incorporation into DIC and biomass in those types of deployments.
Okay, great. And we have time for one last question. What's the difference between a biotrap and a bioflow filter?
Bioflow filter is just a filtering device that you're going to use on the surface, and you're going to pump groundwater from the monitoring well through the filter, and then that filter is then going to be sent in for analysis. And so once that filter is analyzed, usually by qPCR, if you know the volume of groundwater that you filtered, then you can determine what that snapshot concentrations of those various organisms were per unit volume in that well. The biotrap, on the other hand, is going to be incubated in the groundwater monitoring well typically for 30 days, allowing the organisms that are present and active in the aquifer to come into equilibrium with biofilms that are forming on the inside of the beads. And then that biotrap is going to be pulled out of the groundwater monitoring well for analysis, giving you a more integrated look at what the microbiology of that environment is.
Okay, great. All right. Well, thank you very much, Carrie, for this webinar. It's been a great webinar as always. And if we have additional questions that come in, we'll put together a little document and circulate it around to everyone with answers to the questions that are being addressed. But thank you very much, Carrie, for your time, and thank you everyone for joining us for our webinar this month. We really appreciate you being here, and thanks again, Carrie.
All right. Thank you. Bye-bye.
All right. Bye-bye.